thank you again for accepting our invitation, because this time is very, it's a difficult time for us. The vaccination law is already in the parliament. So Dr. James, first tell us a little bit about you, your background, your education.
Okay, I'd be happy to. Let me see. So I've been in biomedical research for 18 years. I was faculty in the Department of Pathology at the University of Pittsburgh and at the University of Pittsburgh Cancer Institute until 2014. I am an expert in genomics, proteomics, bioinformatics, and cancer research and other diseases. I focus on molecular and cellular basis of disease. I designed over 100 research studies at the University of Pittsburgh as the director of their bioinformatics analysis core. I have over 57 peer-reviewed research studies. I was founding editor-in-chief in Cancer Informatics, a journal, and now I am editor-in-chief in Science, Public Health Policy, and the Law. This is the screenshot of the journal Science, Public Health Policy, and the Law. The papers are position papers that focus on the mismatch between public health policy, law, and science. Okay, that's the first question.
I also know that you started studying in the biology and even zoology field. Is this true?
Correct. I have a start in biology, and I have a PhD in ecology, evolution, and conservation biology. I moved towards biomedical research when I was a postdoc at Penn State University.
Dr. James, what do you think this biology background have brought to you? What would be this advantage, let's say, comparing to a usual doctor?
I think the number one most important attribute of my training that prepared me to take on the role that I play now in American society is a firm foundation in scientific research, specifically the principles of objective scientific research. And secondarily, I also have a broad interest in medicine and the pathophysiology or the molecular causes of disease, because I'm not a medical specialist and I have collaborated on over 100 studies. I have a very broad experience in biomedicine. So combining those two placed me in a role where the University of Pittsburgh trusted me with most of their bioinformatics and genomics research.
So our next question for you, Dr. James, is how did you get involved into this vaccination issue?
Yeah, while I was at the University of Pittsburgh, there was no vaccine research that I was aware of that was going on. All of the vaccine safety research was cloistered in the U.S. Centers of Disease Control, and they contracted to scientists in Denmark. So when I decided to write a book, Cures versus Profits, focused on many different aspects of biomedical research that was designed for the public to read to understand what good biomedical research is, I had written 17 chapters on many different topics and then added a chapter on vaccines. In reading the vaccine safety studies, I was surprised to see how poorly the vaccine safety studies were conducted. And I identified 4 major controversies in vaccine safety science. Among these were the question on whether vaccines cause autism. I found that the CDC had not conducted sufficient studies to conclude that all vaccines do not cause autism. They had only focused on 2 vaccines and thimerosal. And so I decided to publish the chapter and reveal the 4 major controversies that are still unresolved. There was a whistleblower at the CDC named William Thompson who stepped forward and said that they had hidden results that the MMR vaccine may be associated with autism. Okay, so given there was not a fundamental basis for this massive, important claim that vaccines do not cause autism, there wasn't a strong basis for this claim. I then decided to read 2,000 studies on autism to determine for myself if there was a plausibility that vaccines might be involved in autism.
In what year did you write this book?
2015. So this. Let me stop sharing for a minute. 2015 was when Cures versus Profits came out.
Okay, so it was.
And the next year, I wrote this book. 2016, okay. I think it came out.
So you wrote Cures versus Profits after the story of William Thompson.
Before, yeah. Well, it was during the story. It was right after Brian Hooker released the transcripts. I was writing the book at that time. And so I decided to write a definitive book on all causes of autism, and the title is therefore The Environmental and Genetic Causes of Autism. I included all of the chemical exposures and the science and the studies of chemical exposures. I included all the genetic studies, and I determined that autism is what I call an acquired cellular detoxification deficiency syndrome. So that's the end of that question. That's how I got involved. Okay.
Oh, okay. Okay, and now we are coming to the Institute for Pure and Applied Knowledge. When did you found this, and why?
So I created the Institute for Pure and Applied Knowledge because I realized that the world needed an independent research institute that could not be touched by corporate profit motive. It was created in late 2015, and we began research activities in 2016. We only accept donations from the public, and we never accept corporate funding or funding from any government agency.
Well done. Congratulations, Dr. James.
Thank you. So that's the story of IPAC.
Okay, and what are you studying there now? So in the present time.
We have studied a number of topics, the first of which I'll discuss today. Is aluminum toxicity. The U.S. Federal Food and Drug Administration has never published a pediatric dose limit of aluminum in vaccines. In our first study, we established an estimated pediatric dose limit for aluminum in vaccines. In that study, we demonstrated that the doses of aluminum from pediatric vaccines implied acute toxicity. In our second study, we determined that the Cdc schedule causes us to expect prolonged and chronic toxicity. We also found that infants that are vaccinated on the CDC schedule receive far more aluminum from vaccines than from breast milk formula or water, if you consider body weight and low absorption of injected aluminum from food and water. Infants,
but these are actually totally different forms of aluminium. One is injected, one is breast milk.
That's an excellent point, and that's part of why I'm doing what I'm doing. The FDA bases all of their safety studies for any dose of aluminum in vaccines on oral doses fed to mice and rats. We found in our second study that infants are expected to be in aluminum toxicity from vaccines 70% of their days in the first 7 months of life.
So can you explain a little more? What does this mean, 70%?
I will. I have slides on it, yes. This is just the introduction, so I'll explain more in a moment. We found that infants are in aluminum toxicity from vaccines 1 out of 4 days in the first 2 years of life.
What do you mean 1 in 4 days?
I'll explain that. I have slides on that.
Okay.
Yeah. So these are the two studies. They both survived blinded peer review. I didn't know who the reviewers were, and they're published in the Journal of Trace Elements of Medicine and Biology. Given the doses of aluminum per vaccine on the CDC schedule that's used in the United States and elsewhere, we determined not merely the total exposure, but also considered the clearance rates.
So you also consider the capacity of detoxifying from aluminium, of spontaneous detoxification.
Yes.
Okay.
I'm about to show you that. Yes. FDA has published an 850 microgram per dose of aluminum in vaccines.
Has published?
The FDA has published a regulation of 850 micrograms per dose of vaccine. It should be expressed not per dose, but it should be expressed per microgram per kilogram per day. When you correct for body weight, these lines represent the pediatric dose limit that we think should apply to aluminum. We published this in 2018. Okay, so I'm going to skip ahead here. When you consider the clearance and the repeated exposure, in the first 6 months of life, we see at least 4 acute episodes of toxicity, even considering the FDA limit.
And in the first 6 months, right? You said.
So I'm sorry, that's in the first 2 years. So that's in the first 2 years. But this 850 microgram limit is meaningless, and it applies to adults. This is the pediatric dose limit, and you can see that there's many days that the aluminum microgram per day exceeds the pediatric dose limit even after the vaccination when clearance has started. Okay, so in 2018, we looked at the doses and exposures once adjusted for body weight from vaccines, and we found that, and it's right on the screen here, in a male child from birth to 36 months, 50 percentile body weight, the FDA dose of 850 micrograms reveals the black line is the safe limit that we expect to apply, and these lines are the exposures from the 3 different doses of aluminum. And at birth, the exposure is clearly very toxic, and as the children age and they increase in body weight until they become an adult, the relationship then inverts when you become an adult. So this is just demonstrating mathematically the exposure far exceeds a body weight corrected toxic level.
But Dr. James, are there vaccines at birth in the United States?
On first day of life, they receive 250 micrograms of aluminum and hepatitis B.
Okay. I asked because I thought you do not have this vaccination at birth. Some countries do not have any vaccine at birth.
Unfortunately, we do. 250 micrograms in hepatitis B vaccine, day 1.
We have the same vaccination in Romania. Plus BCG. I know in the United States you don't have the BCG, Bacille calmette-guerin. It's the vaccine against tuberculosis. We still have it in Romania, and it's given in day 2-3 of life. So here they receive even more than hepatitis B vaccine. We already have a question by one of the participant doctors. She asks if the absorption of the aluminum from injections from vaccines is bigger than the digestive absorption, because many pro-vaccine doctors argue that the absorption from the injectable aluminium is much less than the digestive route.
Yes, let me show you in the appendix in our study. I'll go right to the study and I'll show you that we published this. Once you correct for body weight, and we only absorb 0.3% of aluminum from diet. In the first 6 months of life, we see far more aluminum from injected from vaccines than from food exposure.
But I know there was a controversy that actually the aluminum from the vaccine stays in the muscle where it has been injected, and it is not absorbed in the blood.
I'll discuss that in a moment. First, I want to show the data. So the raw intake from breast milk, parenteral nutrition, infant formula, and soy-based formula in terms of exposure is higher if it's injected compared to vaccines.
Sorry, I don't understand.
The raw exposure, I have a two-part answer. The first part is, yes, on raw exposure of ingested versus injected, there's more in breast milk, parental nutrition, and infant formula, and soy-based formula than it is for vaccine in the first 6 months of life. This is the total exposure first 6 months of life.
So you mean in absolute numbers?
Correct. But we know from this research right here, humans only absorb 0.3% of the aluminum that we eat. Here. Okay, right. There. See, this is breast milk, parenteral nutrition, infant formula, soy-based formula, and vaccines. So the raw exposure is higher from these sources, yes. But we know from food science research that we only absorb 0.3% of the aluminum we ingest. The part two is since we only absorb 0.3% of the aluminum that we ingest, it's an inverse relationship now in the first 6 months of life, and you get far more aluminum into the blood from vaccines than you do from food.
Okay.
Now go ahead and ask your question about the aluminum making it into the blood from vaccines.
And maybe also the form of aluminium that exists in the vaccines, it's a different chemical form than the aluminium in the food. This comes to me intuitively.
So my answer to that is I agree. However, my studies are a response to FDA, who made the comparison by using animal studies of ingested forms of aluminum to assess safety for injected forms. And I'm responding to the claims that you get more from vaccine, that you get more from food than you get from vaccines. And I'm showing them mathematically that is an incorrect assertion. I have another part to this answer.
Okay.
You had also asked earlier about the absorption from vaccines, that it stays in the muscle, and it cannot be detected in plasma or blood. And the problem with that perspective is we don't care about aluminum that's in plasma or blood. We care about the aluminum that's in our tissues. And we know that it gets distributed to the bone and the brain and other organs.
So bones, brain?
Yes. Now we have some preliminary data from vaccinated versus unvaccinated people in children in a pediatric practice in the United States that show increased risk, very high increased risk of anemia.
Yes.
The aluminum is known to bind to transferrin, a protein that captures iron from the diet and puts it in the bones to make red blood cells. Okay. When aluminum takes the place of iron and goes to the bones can't make, in the bone marrow, the bones can't make the red blood cells. I think this explains the high level of anemia. Also, many parts of the of the immune system are active in the bone. And it's a myth that it is safe because we can't detect it in plasma. It is terrifying that we can't detect it in plasma. It means it's doing damage to human tissue. Okay, so in our second study, we compared the acute exposure and chronic retention of aluminum in 3 vaccine schedules. The first schedule is the CDC schedule. The second schedule was a modified CDC schedule that used commercially available vaccines with no aluminum. And the third schedule is called the Vaccine-Friendly Plan by Dr. Paul Thomas. The clearance model that we used was based on injected aluminum hydroxide in adults. And we know that infants have at birth a 20% glomerular filtration rate. Their kidneys work at 20% of efficiency compared to 2-year-olds. So why would we be concerned about aluminum? Aluminum is found in amyloid. It's part of amyloid. It's a protein, it's a non-protein part of amyloid in Alzheimer's brain and in the brains of children with autism. We also know that we can use aluminum hydroxide to routinely and reliably induce autoimmune conditions in animals, in mice and rats. It has other effects as well. We discussed RBC, red blood cell production, but it also inhibits defensive mechanisms connected with white blood cells and macrophages. Aluminum also causes endoplasmic reticulum stress, cell death in neuroblastoma cell lines. It also perturbs cytoskeleton dynamics in human astrocytoma cells. It is a myth that aluminum cannot cross the blood-brain barrier. There are many studies that show aluminum toxicity. In particular, there are many studies that use aluminum to cause autoimmunity in mice and rats. The form of aluminum used in these studies is aluminum hydroxide, the same form as is found in vaccines. And my calculations show that the amount of aluminum in the CDC schedule overlaps the amount of aluminum that these mice are exposed to cause these autoimmune conditions such as atherosclerosis, lupus, allergic rhinitis, asthma, etc. The FDA based their 850 microgram level on one study, this study by Golub et al. They specifically chose this study because it had the fewest adverse events associated with ingested forms of aluminum, so Anita's concern is well placed. They should not use ingested forms of aluminum to assess safety of injected forms. A more recent study with the same dose found neuroinflammation, loss of dendritic spines, and something called immunoneuroexcitotoxicity. They ignored other studies. They have ignored other studies that have had used lower doses that had serious outcomes, which you can read here and then translate as you say them. This again is the clearance graph from our publications. And I'm going to show you how we calculated the chronic toxicity. We calculated the percentage of the days in each week of life that a child can be expected to be over the pediatric dose limit. This part of the graph, part A, is the CDC schedule, and you can see long periods of time where the child is in aluminum toxicity. This is the modified CDC schedule that trades non-aluminum containing vaccines for aluminum containing vaccines whenever possible. This is the vaccine-friendly plan exposure. In this schedule, he skips a few aluminum containing vaccines and uses non-aluminum containing vaccines whenever possible. So for instance, during pregnancy, he does no vaccines. At birth, he does no hepatitis B. He doesn't offer the chickenpox vaccine.
Well, is there a vaccine? Are there vaccines recommended in pregnancy in the United States? In Romania, we still don't have vaccines. I mean, compulsory vaccines for pregnant women.
Yes, only 20 states require vaccination to go to school, and you can receive the vaccines in pharmacy. Yes.
No, I mean for the pregnant women.
Oh, in pregnant women, yes, it's recommended. The CDC recommends vaccination for pregnant women, and there were no safety studies based on this whatsoever.
What vaccines do they recommend to the?
The CDC recommends the influenza vaccine, which can contain thimerosal, a source of mercury, and they recommend the Tdap vaccine to fight against pertussis, which contains a large amount of aluminum. So this is the comparison of the percentage of days that are children in the first 7 months of life are over the pediatric dose limit. The CDC schedule, we can expect that infants in the first 7 months of life are spending 70% of their time in aluminum toxicity. In the modified plan, it's 25%. The vaccine-friendly plan, it's about 5%. In the first 2 years of life, children on the CDC schedule can be expected to be in aluminum toxicity 1 out of 4 days, 25% of their days. The other two schedules are obviously lower, with the vaccine-friendly plan being the lowest. So we recommend that science be conducted to perform per body weight dose escalation studies of aluminum in mice. That study has never been done. We need to reevaluate the use of aluminum in vaccines altogether. And we need to encourage awareness of the differences between vaccine risk awareness, which can lead to improvements in vaccines, and anti-vaccination positions. This leads to the question of international consensus on informed consent. Individuals are entitled to full, free, and prior informed consent for any medical procedure. Individuals must not be experimented upon without their expressed informed consent. This was decided at Nuremberg. It is the policy of the World Health Organization, and it is the policy of UNESCO. You wanted to know what I knew about autism, so part of our research is on autism. In that book that I mentioned, I read 2,000 studies on autism to assess the molecular and cellular plausibility. I concluded that genes and environmental interact. The scientific literature on mercury has been misinterpreted. There is more mercury in the brain and longer from vaccines than from other sources. And remember, I found no studies that have been conducted that compare fully vaccinated to unvaccinated children. And all studies cited by the CDC on the question of vaccines and autism focus on MMR, the measles, mumps, and rubella, and thimerosal-containing vaccines only. In my deep dive in the literature, I did not find studies that exonerated vaccines, but I did find some studies that implicated some of these vaccines.
Yes.
And yet the CDC website claims that vaccines do not cause autism. Because no studies exist for rotavirus, pneumococcal conjugate, inactivated poliovirus, meningococcal, and other vaccines, we must be forced to conclude that the CDC uses magic to determine that vaccines do not cause autism. So our present research at IPAC includes autism, a study of vaccinated versus unvaccinated health outcomes, and coronavirus. You can read about autism as an acquired cellular detoxification deficiency syndrome in my open access paper, peer-reviewed.
Okay, and will you, this is in your PowerPoint presentation that you will send to
The
email?
PowerPoint, yes, and absolutely. This is available freely online, this paper, yeah. Mm-hmm.
Okay.
And if you want to help support IPAC in these efforts, visit our website. We encourage small monthly donations rather than a one-time donation, but we're not tax deductible in the US.
Okay.
Thank you for your time.
Thank you very much, Dr. James, for everything you shared. And so you authorized me to send the presentation, your presentation. And I asked you in the email if you would agree me to record this meeting.
Yes, that's absolutely fine. I will resend the slides because I made some corrections on the fly.
Okay, okay. Thank you very much. Thank you very much, and we'll keep in touch.
You're welcome, and good luck to everyone. Good luck, everyone.
Thank you.
Thank you.
Thank you very much.